So welcome everyone to the NPTEL course of Post-Harvest Operations and Processing of Fruits, Vegetables, Spices and Plantation Crop Products. So this course is instructed by Professor H.N. Mishra from the Department of Agricultural and Food Engineering, IIT Kharagpur, and I am Shikha Sharma. I am a teaching assistant and I am also for this course for July 2025 semester, and I'm a research scholar in the Department of Chemical Engineering, IIT Guwahati. So this live interaction is basically for week 5, uh, and the assignment deadline for week 5 is 27th of August. So this course is basically a discussion regarding all these topics, the module 5, which includes minimal processing strategies, hurdle technology concepts, intermediate and high moisture fruit products, cut fruits and vegetables. So this module 5 is having 5 basically lectures, which includes from 21 to 25. Like for 21 is on minimal processing, lecture 22 is on hurdle technology concepts, lecture 23 is on intermediate and high moisture fruit products. Lecture 24 is about cut fruits and vegetables, and lecture 25 is also on the cut fruits and vegetables. So these are the 5 lecture videos of module 5. I hope you have all gone through these 5 lecture videos. So we can solve some of the concept-based questions, and in today's class, we'll be solving one numerical, and all others will be concept-based question based on these 5 topics. So if you have any doubt from the previous class or any doubts regarding these 5 topics, or from the today's tutorial session, if you have some doubts, please feel free to ask. So let's begin with our first question of today. So question 1 is about which of the following is not a primary factor in hurdle technology? pH, water activity, temperature, color. So which option should be correct? Can anyone share his or her opinion on this question? Which of the following is not a primary factor in hurdle technology? I'm not able to see chat box. Yeah, so Vishakha D. Vishakha is saying D. Okay. So Vishakha is saying D, which is color. Let's see. Yes, she's correct. So color is basically not a primary factor in hurdle technology. So first let's understand what is hurdle technology. So hurdle technology is also known as combined method technology because there is a combination of various methods. So in hurdle technology, basically an intelligent combination we take of various preservation technologies or the techniques or the hurdles. So we are making some combination from the different hurdles. So hurdles can be pH, water activity, or temperature, and we can make a combination of these technologies, preservation technologies. Uh, so we the combination is known as hurdle technology. So the hurdles are chosen carefully in order to maximize the preservative effect. So what whatever we are choosing the preservation methods like pH or water activity or temperature, anything. So what happens? The synergetic effect should be there. Synergistic effect should be there. So what happens is, uh, so uh, we should choose those hurdles, and we should put the hurdles in proper sequence, so the max effect of each preservation technology should be maximized while minimizing the impact on the product quality and product on our and the product quality should be as fresh as possible or as raw as possible. The effect on the product quality or the sensory attributes or its nutritional quality should be minimum, whereas the effect on the microorganism or killing of the microorganism should be maximum, or degrade or any degradative, like on degradative enzymes, the effect should be maximum. Usually, what happens? This is achieved by combining processes with synergistic effect. So, we should choose such processes which are having like 3 to 4 processes which can provide some synergistic effect, so that there uh, we, the amplitude of each treatment, like if we are applying pH, the amplitude and temperature, suppose. So the amplitude will not be much, like the temperature will not be very high and the pH will not be very low, but uh, their effect will be, cumulative effect will be very high. So we need not to apply severe treatments while doing the hurdle technology, while using hurdle technology, and that's why the product quality remains intact in this approach. So these hurdles, they can be any physical, chemical, or biological factors that create an unfavorable environment for the microorganisms or enzymes also, like degradative enzymes we can include in this, while maintaining the product integrity. So product quality should not be affected. It should be as fresh as possible, as raw as possible. Whereas the degradative enzymes or microorganisms or the pathogens, spores, we can include everything like spores, or we can include bacteria, yeast and mold, everything. So they on them the effect will be maximum. They should they should be killed by these combination of the technologies of preservative technology. So in this picture we can see that um, from one hurdle, this is hurdle one. So what happens? This microorganism is able to cope up with this and can this hurdle is not affecting its growth. Whereas if we see the second hurdle, this is the barrier. So microorganism will not be able to cross this. But in hurdle technology, we will use multiple hurdles or barriers so that microorganism of small intensity, like this one is first hurdle, this is second hurdle, then this is third hurdle. So microorganism will not able to jump, will not able to grow or survive. Likewise, this combination hurdle technology or theory also known as soft technology, because we are using the intensity of applying of one preservative technology is low compared to when we are applying single, like single, if we are applying temperature, we'll use more higher temperature, or we use very low temperature. Whereas when we are using in combination of the different technologies, so the intensity will be not so severe, not so high. So this is microorganism, harmful microorganism. So we are using gas atmosphere as one of the hurdle. We are limiting oxygen amount, let's suppose. And then in second case, we are using some preservative. Then we are using third case temperature also. In fourth case, we are using pH also. And fifth case, we are using water activity also. So all from all these preservative technologies, this microorganism will not able to grow or survive. So basically the principal hurdles which are there. So the principal hurdles, preservative factors in food safety are basically temperature. So it can be high or low, water activity, then acidity or pH, then redox potential, then chemical preservatives like nitrite, sorbate, sulfite, then vacuum packaging where oxygen is not there, then modified atmosphere packaging where oxygen and CO2 will be in certain amount, HHP which is high hydrostatic pressure, and UV, uh, we know ultraviolet radiation, and then sometimes competitive microorganisms like lactobacillus. So like this, we can use several preservative these hurdles to uh, control the growth of the spoiling microorganism. Whereas color is basically not a primary factor. So it is basically related to the sensory attributes of the food products. Basically hurdles, they can be classified into physical hurdles where like temperature can be there, and physiochemical, which means physical plus chemical, right? So it includes pH, redox potential, etc. Then non-thermal methods are also there, like high hydrostatic pressure or pulse electric field or pulse light, ohmic heating. But ohmic heating will come in the like thermal treatments, not in the non-thermal. So another are microbiological. So we can use desired competitive microflora in the product so that particular microorganism are desired, but they'll give competition to the spoiling microorganisms. So this is required this is required one, this is not required, right? So this can give competition to the spoiling or the spoiling microorganisms, spoilage microorganisms or pathogens. So the color is not a primary factor in hurdle technology. Let's move to the second question. Which of the following statement are true regarding the principles of hurdle technology? So, first one is hurdles disturb the homeostasis of microorganisms. Second, microorganisms can easily jump over multiple hurdles. Third, hurdles can cause microorganisms to remain inactive or even die. Fourth, combination of hurdles prevents microbial growth. So, which statement are true? Suraj is saying A, 1, 3, 4. Vishakha is also saying A. Okay, guys, let's see answer. So you guys are correct. A is correct answer. So we just now understood that what are basically hurdles. So we need to tell the principle of hurdle technology. So basically hurdles, they are applied to the product for the disturbance of the homeostasis of microorganism. So statement 1 is correct that hurdles disturbs homeostasis of microorganisms. Then statement second says microorganisms are not able to jump over all the hurdles present in the food. So obviously microorganisms will, so we can see from this picture that this is first hurdle is temperature, second is water activity, third is pH, fourth one is redox potential, and then fifth one is pressure. So we can see that microorganisms have initially very much high power, so it can easily cross this first hurdle, then second hurdle, then the microorganism has little less energy, but it will still be able to cross third hurdle. Then it is having more less energy, but still it can manage to cross fourth hurdle. But at fifth hurdle, it is completely exhausted and cannot be able to cross. So this by this figure we can understand that how hurdle technology works. So statement 2 is incorrect that microorganism can easily jump over multiple hurdles. No, it cannot be able to jump over all the hurdles. So this one is correct. Sorry, incorrect. Then third is hurdles cause microorganisms to remain inactive or even die. So basically, and also fourth is saying that combination of hurdles prevent microbial growth. So obviously, these two are correct. So preservative factors prevent microorganisms from multiplying and causing them to remain inactive or even die. So these hurdles will inactivate or even help to inhibit the growth of microorganisms. And in hurdle technology, we have combination of hurdles. So this figure well depicts this part, and A is the correct answer. So if we talk about the in the hurdle technology, so there are basically four mechanisms by which this microorganism is going to kill or its growth will going to be inhibited. So first one is homeostasis. So we are disturbing basically homeostasis of microorganisms. What we are doing is homeostasis is basically tendency to maintain uniformity and stability in the internal microbial cells, which is essential for its survival. So basically, this is microbial cell. So it has certain condition in its internal cell. This internal cell will have certain condition, like it will have certain amount of pH or certain amount of redox potential or anything. So these parameters will be in certain amount so that the function of the microbial cell will be regular. So there may be it like the micro, because if the maybe the microorganism can grow at neutral pH, maybe around 7. So if the pH will be very low, so what will going to happen? The proteins which are working in the cell of the microorganisms, they will denature and lose their functionality. So this pH is important or mandatory for bacteria for its growth and the survival. So if so homeostasis word itself says that the microorganisms are able to maintain the uniform or the equilibrium, or what we can say neutral condition or best condition for its survival in the internal microbial cells, which is essential for its survival. So if the microorganism will be in its uh, best or optimal condition of growth, it means it is in its homeostasis condition. So for its uh, inhibition, we should disturb this condition of homeostasis. So once the homeostasis causing microorganisms disturb causing microorganism disturbance, if there is this disturbance in the homeostasis, so what happens through the maybe incorporation of any hurdle, so we are applying hurdle to disturb this homeostasis condition, then the microbial spoilage will either be delayed or the microorganism will be killed. So microorganism can be killed by its growth can be inhibited or it can be killed. So we can have several stress factors or hurdles like low level of the nutrients or low pH, low water activity, low temperature, rising level of oxygen, then presence of biocides, then ionizing radiations, then competitive microflora. So these are the different hurdles which can basically disturb the homeostasis. So because of that, homeostasis response will be there in microbial cell. Like if we talk about just one example, so if the low level of nutrients will be there, so what happened? Low level of the metabolism is there. So more the microbial will undergo in the stationary phase. It will not grow properly, right? So if we talk about the pH, if pH is low, so what happens? More H plus ion will be there inside the cell. So removal of the protons from the cell. So these protons should be removed outside the cell so that pH can be maintained. So like these are the different stress factors which disturbs the homeostasis of microbial cell. The second mechanism is metabolic exhaustion or auto-sterilization. So what happens is this is the first mechanism in the cell uh, for hurdle technology. This is the second mechanism which says about first the hemostasis. If it is disturbed, then what happens? In hurdle treated products, microorganism it basically uses its energy to maintain the hemostasis condition because we are disturbing the hemostasis in first case. So for maintaining this hemostasis condition, microorganism requires energy. So in the end, microbes will be metabolically exhausted. So if so many hurdles will be there, then microorganism will be exhausted, its energy will go, and eventually the microorganism will die because of starvation, because there is no energy. So this process is called auto-sterilization. So because microorganism is going to kill, so because of the energy exhaustion, so auto-sterilization, we term this as. So auto-sterilization provides food safety against pathogens and spoilers during storage, mostly at room temperature. So if we have small intensity hurdles, so what will happen? Microorganism energy will go and it will be metabolically exhausted and die so that our food will be in good condition. So this diagram is these two diagrams, they both are for depicting the pH. Like if the pH is very low outside, so what happens from the outer membrane to inner membrane, this pH H plus or ions will be leaked. So if the pH, suppose in the uh, outside is low, so what will happen? Inside pH will be like different and outside pH will be very low. So the H plus ion can come from higher concentration to lower, which is from outside to inside. So because H plus ion is there, so what happens? Microbial cell will try to remove this H plus ion and take some maybe Cl minus or minus ions so that the pH can be balanced. It can undergo also in some cases, uh, what happened, it can undergo in the decarboxylation process, or the decarboxylation. So what will going to happen, uh, like the H plus ion should be consumed. So what will happen in this reaction? Uh, so this product RH will form from this reaction and CO2 will be released. So CO2 will go out, RH will go out from the cell's ports, and that's in like the byproduct of RH, the byproduct of H plus ion, like RH, R negative H plus. So it will go out. So this way, cell will try its best to remove all the H plus ions. Or what next can do, like ADP will be converted into ATP, because ADP and NADH also will convert into NADH. So all these things, all the reactions which requires H plus ions, they will start and try to reduce the number of H plus ions. After that, what will happen? Stress reaction will be there. So the stress reaction has been observed in microorganisms when they are under stress due to food shortage. So microorganisms, they become more resistant under the stress, and they will release stress shock proteins. The rate of the formation of the stress shock proteins, they will depend on the pH, water activity, ethanol, and heat. The synthesis of shock proteins become difficult when more hurdles are introduced at the same time and can lead to metabolic exhaustion of microorganisms. So to combat with the situation, microorganisms and first microorganisms hemostasis is disturbed. After that, the energy or the metabolic exhaustion will be there because energy will be low. And if combination of hurdles will be there, what will going to happen? And food is not there, so microorganism is under so much of stress. In that condition, it releases stress shock proteins, and because microorganism is synthesizing stress shock proteins, so it needs more energy, but the energy is very low. So further metabolic exhaustion condition is there for microorganism. So last is the multiple target preservation of food. So the combination of these different hurdles, like we are applying pH, water activity, and redox potential, etc. So they will act synergistically on microbial cell. So microorganism will not able to basically survive. So the multiple hurdles, they will simultaneously target different cellular components, such as one is targeting cell membrane, so cell membrane integrity, another is targeting enzyme function, then another is targeting DNA stability, then another hurdle will target pH homeostasis, and another the water activity will target water activity levels or low levels of water activity. So all these multiple hurdles will going to act synergistically and disturb the homeostasis of microorganism and reduces the synthesis of stress shock proteins. So if we apply all these hurdles simultaneously, so microorganism will have different stresses. So it will reduce its energy sharply, So the synergistic effect of different preservative factors or the hurdles which are having even just a small intensity, they'll be more effective on the microbial cell than one preservative factor of larger intensity. So what will going to happen? These small intensity hurdles, like we can see in diagram also, that microorganism listeria we are taking. So many small hurdles are there, but of small intensity. So they'll act simultaneously and will show synergistic effect, and microorganism will not able to survive. It will be killed, and the product quality will be as fresh as possible. So let's see this question number 3. Match the hurdles which are given in column 1, like low pH, high temperature, low water activity, and blanching. In column 2, we have their effects like inhibit microbial growth, causes metabolic exhaustion. Third is stress reaction in microorganisms, and fourth is disturbs homeostasis. We need to match this. Anyone would like to answer? So is there any doubt so far in the maybe in the previous question? Anyone who would like to help me with this match? The following. Suraj is saying D. Okay, let's see the answer. Suraj answer is A. Let's understand why. So basically low pH is basically first in it is inhibiting microbial growth, and then high temperature is basically disturbing the homeostasis, and then low water activity causes the metabolic exhaustion, and further if we are applying blanching, it is reducing stress reaction in microorganisms. So if we talk about particular uh, if we talk about one hurdle, particular hurdles, like first is food pH, like the low pH. So what happens? The free H plus ion is affecting the microorganism growth. So if its concentration is high, and if free H plus ions concentration is high, or if there is a good amount of weak acid present in the food, so weak acids they will dissociate and produce free H plus ions. So weak acids, they will cause leakage of H plus ions across the cell membrane of the microorganism, which will going to acidify the cell interior. So higher will be amount of H plus ion. So it will come inside the cell and it will acidify, and it will inhibit transport mechanism of the microorganism. So growth of the microorganism will be inhibited, and there will be like what there will be uh, problem, or there will be interference with the cell membrane, because like there will be some dysfunction in the cell membrane, or metabolic enzyme activities, protein synthesis system, and or the genetic system, genetic material. So it will this low pH or the acid environment will affect all these primary metabolism in the microorganism. So in a mixed initial flora, The acidity and pH of the food may determine the potential spoilage microorganism. So we can, if we have many spoilage microorganism, we can find one target microorganism and apply the pH according to that. Like pathogens, if we talk, and spores, we can talk. So pathogen cannot grow at low pH or pH less than 4.5, like in juices and the sauerkrauts. And spores will also not be able to germinate. So we'll take the we'll take the target microorganism and apply the hurdle according to that. So in this, if the pH is coming, uh, if the pH is low, so H plus ion is coming inside and making this internal environment also acidifying. So what will happen? Microorganism will try to go in the amino acid decarboxylase system and try to remove the H plus in with RH, and also some buffering ions, like it can use like Cl minus, and sometimes it will exclude, or it will it will take out all the plus like plus one ions, because H plus is already there. So Na plus or K plus ion can be thrown out of the cell, so that the concentration of the ions can be equilibrated. When the water activity will be low, so what will happen is microorganisms are metabolically active only in certain range of water activities. So microorganisms do not contain water impermeable barriers. So microorganisms, in their cell wall, there is no water impermeable barriers. So what will happen if the low water is outside? So the turgor pressure inside the cell will also be affected. So what will happen if low water activity is outside? So water will go from higher concentration to lower. So the cell water will go out, so and tend rapidly into osmotic equilibration with the surrounding. So if water is low outside, so by the principle of osmotic equilibration, cell will loses water and itself shrinks, like this diagram. So at low water activity, the osmolarity of the environment, it will be high, and the solutes present cannot penetrate the cell membrane, and the microorganisms, it will going to lose the water. So the loss of the water, it will result in the reduced metabolic activity, and temporarily the growth of the microorganism ceases, and overall it affects the hemostasis of the cell. So what will going to happen is, if the water is low outside, so to maintain the osmolarity or osmotic equilibrium, the cell water will from, because cell water is more, so it will go out from higher concentration to the lower concentration which is in the surrounding. So since water is going out, so the cell wall is getting in the shrinkage form. So the cytoplasmic volume, cytoplasmic volume of, for the cell cytoplasm volume, it is going to decrease, because the cell is going in the shrinkage form. So because it is going in the shrinkage form, the lysis can also occur in the cell wall. So this stage is known as plasmolysis, where the plasma of the cell is going to be in lysis form. So because of shrinkage, the volume of this plasma is also less, and this certain stage is known as plasmolysis. So because to combat this particular situation, what will going to happen is uh, some like this K plus ion or Na plus ion or the solutes microorganism will remove this or throw out of the cell, so that the cell volume, the inside volume of the cytoplasm can be maintained. Also, it can use certain compatible solutes like glycerols or sorbitols or like erythritol or like these, which is in the fungal cells, and in bacterial cells like proline, glutamic acid, all these aminobutyric acid, these compatible solutes, it will try to attract inside the cell, so that the overall cytoplasmic volume can be remake or can be re-achieved. Next is the temperature. So in temperature, there can be high temperature and low temperature. So right now we are talking about high temperature. So blanching is one of the treatment. So an early processing step is we use blanching as a primary processing step. So this can be used for the destruction of the contaminating microorganisms, the water activity and the pH. Basically, with the blanching, it can reduce initial microbial count, disturbs the hemostasis, and basically finally achieves the microbial stability. So it can decrease microbial viable number of counts if we are applying water activity, pH, and like blanching, all these preservative techniques one at a time. So in that particular process, if we are applying certain processes, so initially from so what we are doing is we are applying several methods. So first homeostasis is disturbed, like homeostasis is disturbed. After that, metabolic exhaustion will be there, and then shock proteins will release, right? So in when we are applying temperature, high temperature, so because of the metabolic exhaustion, auto sterilization is going to happen in the food product. So microorganisms basically exert every possible repair mechanism so that it will overcome from the a certain this stress environment. So it will going to use up its complete energy and become done. So microorganism is metabolically exhausted and it is going to die because of several hurdles we are applying in combination, right? So the high treatment processed fruits, they will become microbially stable even in non-refrigerated condition for several months, because we are applying first, first we are applying suppose water activity, then pH, and then temperature for blanching. So microorganism at the end will become metabolically exhausted. So answer is A here. Let's discuss next question. Which of the following statements are true regarding the effect of hurdles on microbial cell? So first is low water activity leads to metabolic exhaustion. Low pH causes leakage of H plus ions. High temperature increases the production of stress shock proteins. And fourth is blanching removes microbial spores. So anyone? Vishakha 1, 2, 3. A. Okay. Anyone else? Prachi? A. Okay. So let's see. D is correct answer in this question. Let's understand why. So we discussed about many hurdles and their basic principle of like how they are acting or what is their mechanism. So we know that low water activity can lead to metabolic exhaustion, and if low pH is there, so H plus ion will be leaked, right? But why statement 3 is not true, that high temperature increases the production of stress shock proteins and blanching removes microbial spores. So microbial spores cannot be removed by blanching. We know that, okay, because spores uh, like blanching can only inactivate them. And high temperature, it will not uh, increase stress shock proteins, but cause damage to microbial cell. Like it will, if the high temperature will be there, so what will happen? Protein denaturation will be happening. So, or whatever the cell material which is made up of the protein or other things, they will denature. So, obviously, not increase in stress shock proteins, but there will be sorry, damage to the microbial cells. Is this question clear to everyone? Because at high temperature, microbial cell obviously will going to damage, but how can at high temperature proteins formation can happen? No, protein formation cannot happen. So this is not true. Let's move to next question. Which of the following statement is true about the combination of non-thermal hurdles? So we talked about some conventional hurdles before. Now we are talking about non-thermal hurdles. So if we are combining non-thermal hurdles, so it can enhance microbial inactivation. Second statement is saying non-thermal hurdles can be combined with thermal hurdles. Third statement is saying non-thermal hurdles alone are always sufficient for microbial inactivation. Fourth is combination of hurdles can reduce the intensity needed for each hurdle. So Suraj is saying B, 2 and 4. Okay, C, 1, 2, 4. 1, 2, 4, Vishakha also. Okay. So you guys are correct. 1, 2, 4 is correct answer. So when we are combining the non-thermal hurdles, so it will lead to inactivation of microbial microbial cells. So if we are even combining the thermal hurdles, like many non-thermal hurdles with thermal hurdles, or many hurdles or many preservative factors together, so obviously there will be the inactivation of microbial cells. Then second statement is non-thermal hurdles. They obviously can be combined with temperature or thermal hurdles, which is high temperature or blanching or canning, anything. So they can be combined. Then third is that the combination of hurdles, if we are applying in low intensity, like if we are taking combination of hurdles, low intensity, we can reduce the intensity of one hurdle. Obviously, it is true. That's why we are using hurdle technology, because we want to apply low intensity treatment, so that, or low intensity hurdle, so that the microorganism can be killed, but the product quality can remain intact. Then non-thermal hurdles, basically, obviously, they are sometimes not sufficient, are not always sufficient for microbial inactivation. So third statement is wrong, all are correct. So we know that there are different hurdles, like physical, so it can have high temperature, blanching, pasteurization. Low temperature includes chilling, freezing. Electromagnetic energy is also like can use for like microwave and radiation. And sometimes packaging can also act as a hurdle, like active packaging or vacuum packaging, edible packaging, or modified atmosphere packaging, MAP. Then there are different physiochemical hurdles like CO2, O2, O3, ethanol, and then lactic acid, or low pH or low water activity, low redox potential. Then organic acids can be used. Sometimes salt can be used, which is a conventional method. Some spices and herbs can be used. Certain chemicals like sulfides or nitrites can be used. So these can be used. And then non-thermal methods includes all the novel technologies like HPP or HHP, then UV, then pulsed electric field, PEF, and then pulse light, plasma also, one of the thermal approach. And then microbial uh, approach can be used, like competitive microorganisms we can use, so that the uh, it can compete with the uh, undesired or the spoilage causing or the pathogen causing microorganisms, so that the desired, like for the food, if this competition will competitive microorganisms will compete for the food, so the undesired microorganism will feel competition and shortage of food and cannot grow. So that way also they can be used as a one of the hurdles. So next question, let's see which of the following statement is true regarding the high hydrostatic pressure HHP as a non-thermal hurdle. HHP is a combination with antimicrobials to increase effectiveness. HHP alone can inactivate all type of microorganisms. HHP combined with heat reduces processing time. So anyone would like to answer? So till now, all the questions are fine with you. Do you have any doubts so far? If not, please try to give answer of this question. So Vishakha is saying 1 and 3, option A. Suraj is also saying option first. Okay, let's see. So they both are correct. 1 and 3 correct. So what happens? We know that this diagram is for HHP. So basically in HHP, what happens is food like this part we should keep our food. So in this part we can keep our food. This is basically a piston or uh, some this can be manual or automatic. So the piston it will deliver its pressure to the pressure medium. So there will be some pressure medium will be there. In that piston will put all the pressure, and because there is pressure medium, so pressure will going to be uniformly distributed and applied on the food product which is kept in this particular area. This is the basic principle of high hydrostatic pressure. So basically food are subjected to pressures basically at higher pressure, which can be in the range of 100 to 1000 megapascal, and the food can be kept with or without packaging material. So we basically, if we are keeping food with packaging material, this packaging material should be flexible, and it can we can consider around 15% it can change its volume, like it can reduce the volume to 15%. So when the pressure is released, so the this flexible packaging can be regain its that final or original shape or size. So on the food which is in the packaging material or without the packaging material, it can be in a pressure vessel which is capable of sustaining that high temperature. So here food will be there with or without packaging material in the uh, package in the pressure medium, and from above piston will apply very high pressure around 1000 to 10,000, around 1000 to 1000 megapascal, and this pressure can be hold from 30 seconds to 30 minutes. This is basically HHP. So pressure transmitting medium can be water with castor oil, silicon oil, glycol, and ethanol. And it works on basic two principles: isostatic pressure. So since we are using here we can see this is the product with the packaging material, and this outside is the pressure medium. So because of this, uniform pressure is being applied. So application of pressure basically is instantaneous and uniform throughout the sample, and it works on Lee-Chatterjee's principle, which says that reaction resulting in a volume change are influenced by pressure application. So volume is being changed, and so what will happen? First, the pressure will apply, so pressure will increase, which is come up time, and then holding pressure will hold for certain time, which is holding, and then decompression at the final step, decompression will be there. So this is the graph for like how HHP is being applied. And if we see this table depicts certain microorganisms like we see Listeria monocytogenes. So it will be like its 5 log reduction in activation will be at 375 megapascal. So all these are the examples of different microorganisms and their 5 log reduction at which pressure. So this much pressure should be applied, or like E. coli, we can see at 680 megapascal, and aureus, S. aureus will be at 700 megapascal. So this much pressure should be applied so that these pathogens can be killed. And if when this is the pressure temperature graphs. So if we see pressure is increasing, so also because of the increasing in pressure, temperature also a little bit increases. Like initially temperature increases and it will maintain like this much of temperature will be at for certain time, and then when decompression will be there, the temperature will also go low. But this temperature is not very high uh, when we are applying the pressure which is very high in the megapascal range. So this high pressure, it will lead to death of the microorganism because there will be permeation. Because what will going to happen? Uh, this if we talk about, this is the cell of the microorganism, so it will permeate permeate. Like what will going to happen? The cell permeability or the cell membrane permeability, it will going to change, or there can be even pores in the microorganism cell membrane due to permeabilization of the cell membrane. So cell membrane, it will it will not be okay. It will be degraded. So because of that, the cell death will be happen. Then changes can be reversible at low pressure and irreversible at high pressures. And there will be like reduction in the enzyme activity when we apply HHP. So which means that there will be high quality and stability of the food product, and mostly non-covalent bonds are affected. So organoleptic property, it will be same. It will not alter. High hydrostatic pressure is basically sometimes can be combined with antimicrobials and heat also, so that the effectiveness of the treatment will increase and its processing time will decrease. So HHP is effective for a good range of microorganisms, but not all the types. So statement 1 is correct and statement 3 is also correct, but second one is not correct because it cannot apply to all microorganisms. Now let's discuss about pulse electric field. So which of the following statement is correct regarding pulse electric field as a non-thermal hurdle method? So statement 1 says high intensity pulse electric field causes change in the cell membrane due to an electromechanical instability. Statement second is saying combination of heat, lysozyme and high intensity pulse electric field are potential alternatives for heat sterilization. Statement 3 says the electric field and ionic strength are not related to the poration rate and physical damage of the cell membrane. Statement 1, fourth says pH changes the cytoplasmic condition due to the osmotic imbalance caused by the poration. So, anyone would like to answer? Sugraj, C. Vishakha, 1, 2, 4. This one. Okay, let's discuss the solution. D is the correct answer. 1, 2, 4. So let's understand what is pulse electric field first, and then we will solve this question. So pulsed electric field is basically one of the non-thermal method of the food preservation, which has what we use is short pulses of electricity in this case to inactivate the microorganism and cause minimal detrimental effect on the food quality attributes. So we uses here short pulses of electricity for microbial inactivation. So when we are exposing a biological cell, be it a plant, animal or microbial cell, to a very high intensity electrical field, so in this case, we go up to 50 kilowatt per centimeter. So when we are using very high intensity electrical field, which is for a very short time, so very short pulses of microsecond to milliseconds, then it induces the formation of temporary or permanent pores on the cell membrane. So what we'll do, we'll take two electrodes of positive and negative side, and if we are putting microbial cell or the we will put our food uh, inside this, and in that microorganisms are also there. So what will going to happen, this is the microorganism cells. So if we are applying very high electric field of short pulses, so what will going to happen, there will be pores. These are the pores which will form in the cell membrane. So this formation of these pores by the help of the electric field is known as electroporation. So it causes the permeabilization of the cell membrane. So cell membrane is going to be like have the pores, and it will be damaged. And if the intensity of the treatment is very high, then cell membrane can also disintegrate. So if uh, E is equals to EC until EC, so there will be some pores. And if the electric field is very high, applied E will be very high. So what will going to happen? This whole cell membrane is disintegrated. This is the pulse electric field. So at high intensity pulse electric field, it changes the cell membrane due to electromechanical instability. So it is correct statement. Then if we are using heat, lysozymes, and high intensity PEF, so it can use uh, for the sterilization and can be used as a potential alternative for the heat sterilization. So this statement is also correct. Second one. Then the electric field or the ionic strength are related to the poration rate and physical damage of the cell membrane. And pH changes the cytoplasmic condition due to the osmotic imbalance, which can also cause, imbalance caused by the poration. So from poration, what can happen? The osmotic imbalance can happen, and pH change can also lead to the uh, condition of the osmotic imbalance. So if we are applying electric field and ionic strength, they are not related. So this is not correct. Related to the poration rate and physical damage to the cell membrane. And pH changes basically cytoplasmic condition. So we discussed in the previous slides that pH will going to change the cytoplasmic condition because H plus ion will be inside, and inside cytoplasm the acidic condition will be there. So because of that, osmotic imbalance will going to happen, and pH changes the cytoplasmic condition. So due to the osmotic imbalance caused by poration. So poration can cause like these two treatments are simultaneously applied, PEF and pulse pH, PEF and pH and pH. So this can cause poration, and pH can then pH can change inside the cytoplasm, and because of these two conditions, the microorganism can die. So B is correct answer here. If we talk about the overall process in the, what will going to happen is from that this diagram in pulse electric field. So if we talk, we have two electrodes. One is negative and another is positive. So if external electric field we are applying, this E is basically we are applying. So what will going to happen? This E will increase so much that the membrane potential, the membrane potential, if the E is more than even the membrane potential, so what is going to happen? This kind of poration will be happening. And if uh, too much electric field, external electric field intensity is very high, so this kind of bigger pores can be formed. So let's discuss this question. Next question, which of the following statement about plasma treatment in food preservation is correct? Statement 1 says plasma treatment uses ionized gas to inactivate microorganisms. Second is it is not effective at low temperature. Plasma treatment is not suitable for solid foods. Fourth is it can improve the shelf life of fresh produce. Anyone would like to answer, or anyone has any doubt so far? Anyone would like to answer this question? B, 2, 3, and 4. Okay, so B and C. Prachi saying B. Okay, we got two answer as B, one as C. So answer B is correct basically. But let's discuss first what is plasma. So plasma is basically fourth state of matter. We have solid state of matter, liquid, gas, and fourth state is plasma. So here the material become like so hot, very hot, like the temperature is so high that electrons basically, these electrons are no longer bound to the individual nuclei. So electrons will going to far away from the nuclei. Plasma basically is an ionized gas which are having free electrons, ions, reactive atoms, neutral molecules, and photons. So basically it can have these basically positive and negative ions, electrons, atoms, free radicals, metastables, photons, all these can be there in plasma. On an average, basically plasma is overall, but overall charge of the plasma is neutral because the same number of positive and same number of negative charges will be there. So overall plasma will be neutral because any charge unbalanced, it will result in the electric field that would tend to move the charges in such way as to neutralize charges of opposite signs. So basically the same number of positive and negative ions will be there in the plasma. So it is overall neutral and it is also known as quasi-neutral gas. Now let's discuss. So basically plasma treatment utilizes ionized gas to inactivate microorganisms, which is correct because it has ions, positive ions, negative ions. So plasma is using ionized gas to inactivate microorganisms. Second is it is not effective at low temperature. So plasma basically uh, is basically effective at low temperature. Not effective. This may, I'm sorry, this is incorrect. Second statement is incorrect because plasma can be effective at low temperature. We have cold plasma and hot plasma, two plasma treatments. So it is effective basically at low temperature. Plasma treatment basically is suitable for both solid and liquid foods. So it's not correct. And it can improve the shelf life of fresh produce. So statement 1 and 4th is correct basically. D is correct, I'm sorry. Because D is correct. Statement B is not correct here. Is this question clear to everyone? Can we move to this next question? Which of the following is a characteristics of intermediate moisture food? Water activity above 0.9, moisture content between 20 to 50%, microbially not stable at room temperature, or low sugar content. Anyone would like to share his or her opinion on this question? B, moisture content between 20 to 50%. Vishakha, you are correct. In intermediate moisture food, basically, the moisture content is around 20 to 50% weight by weight, and they have the water activity between 0.75 to 0.85. So statement 1 is not correct. Then they are generally microbially stable at room temperature. So this is incorrect. Statement C is incorrect. And they often have high sugar content basically. So statement 4th is incorrect. Only statement B is correct. And they can have certain initial characteristics, like they can have certain color, texture, flavor, like a fresh food. And IMF, intermediate moisture foods, they are basically appealed by the consumer, and they have the microbial safety, desired color or desired order, and high nutritional value, and they can be ready to eat. So they have basically the moisture content which is intermediate, like dry foods and high moisture product foods. So they have the intermediate moisture content. Is this question clear to everyone? So there are this certain examples also given in this table which are having certain IMFs. So they have the different water activity, like raisins. It has very low water activity, 0.51 to 0.61, because they are adding sugars and or the sucrose in this, so that this product can be uh, can can have certain moisture but can remain safe. So these kind of food products we can see in the market, like papaya, pineapple, or apple. So these have the high sugar content, so these can be remain safe for longer times. Like we can see that apricots, dried apricots, and peaches can be there. So their water activity will be reduced to 0.73 to 0.81 with the help of sucrose. And in chutney sauce, we are reducing the water activity using NaCl and certain food acids, like up to 0.86. So these are certain examples of IMFs. So these has moisture content little low. Let's discuss this question. What is the water activity range of maple syrup, which is a intermediate moisture food? Anyone would like to answer? In just previous slide, we discussed about certain food products. There it was mentioned, maple syrup also, if you have seen this properly. So maple syrup has a water activity in the range of 0.82 to 0.86. So it is also mentioned here, maple syrup 0.82 to 0.86 by adding the sucrose or other sugars. So maple syrup is basically looks like this little caramel or brown color, like honey. It looks like little like honey. So it is basically a concentrated sweet liquid which can be obtained from sap of maple trees, and basically after taking it, it should go under the process of evaporation of water, so the water content can be removed. So on the basis of how much water we are removing, different uh, grades can be obtained based on the color and the flavor. And raw material is maple syrup basically, which has 2% solids, and it has most of the sucrose, which is in the form of sugar, which is in the form of sucrose. So 97% is sucrose in this, and what this like 2% is solids and 98% is liquid. So that is to be evaporated. So that should be evaporated. So the final concentration of the water can be low. So maple syrup is basically a sweetener and a flavoring agent which can be used in different foods and beverages. And the water activity of the maple syrup is this 0.82 to 0.86. Let's discuss one numerical problem. So in a fruit slurry, if we are talking total moles of the water is 8 moles and moles of the sugar is 4 moles. So what should be the water activity of this fruit slurry? So please solve this question. So we know that the water activity is basically the ratio of the vapor pressure of the food when it is in completely undisturbed balanced form with the surrounding air medium and to the vapor pressure of distilled water under identical condition, P by P naught. And we are here talking about that the ideal solution. So when we are talking about ideal solution, we can use Raoult's law. So Raoult's law is the partial vapor pressure of the component in a solution is directly proportional to its mole fraction x into the vapor pressure of the pure form. So if we talk about P equals to x into P naught, so x, which is mole fraction, is equals to, we can talk P by P naught, which is same as water activity AW. So the water activity of an ideal solution is given by the mole fraction of water. So water activity is nothing but mole fraction when we are talking about ideal solutions. So mole fraction, how we can calculate? Moles of water divide by, moles of water divide by moles of water plus moles of solute or total moles. So moles of water is given as 8. So we can put 8 here, divide by total moles, which is moles of water plus moles of solute. So moles of water is 8 and moles of sugar is 4. So we can put here 4. So 8 divided by 8 plus 4, which is equals to 8 by 12, which is 2 by 3 or 0.67. So answer is B, 0.67. So is this question clear to everyone? Okay, so let's move to this next question. Which of the following method is used for producing IMF? Partial drying, osmotic dehydration, fermentation, vacuum impregnation. Vacuum impregnation. Okay. So in that vacuum should go, like air should, uh, vacuum condition should be maintained. Okay. So which method can be used for making IMF and which cannot be? C, 1, 2, 3. A and C. Okay, let's see answer. So C is correct answer. Fermentation cannot be used for making IMF. So basically, we have different technologies for the production of IMF. Some are conventional, like partial drying, moist infusion, or osmotic dehydration. Then we have dry infusion and like direct formulation. We are making the direct formulation in that particular water activity range. And we have certain novel technologies like innovative drying techniques like microwave, combined microwave and infrared drying. And we have different water activity lowering agents we can use. And osmotic dehydration techniques we can use, vacuum impregnation, in which what will happen, vacuum or air will be removed, so water will also be removed, so with the help with air. Osmodehydrofreezing and pulsed electric field assisted osmotic dehydration and ultrasonic assisted osmotic dehydration. So partial drying also being done as conventional method, osmotic dehydration as innovative method or novel technologies, and also vacuum impregnation, but not fermentation. So C is correct answer. Let's discuss this question. What is typical water activity range for high moisture fluid products? Suraj is saying D. Okay. Others? Is correct. So high moisture fluid products basically are the one which are having water activity in the range of 0.85 to 0.98. So most fresh foods basically they are considered as high moisture foods because and they have more than 50% weight by weight water, and if that water we talk about water activity, it will be very high, like from maybe 0.9 to 0.99, or we can say after 0.85 until 0.98. These foods basically includes beverages, fresh meats, seafood, dairy products, and fruits and vegetables. However, if we talk about intermediate moisture foods, they have water content around 10 to 50% and water activity 0.6 to 0.9, and they includes grains, nuts, dehydrated foods, and many processed foods. Next question, let's discuss about high moisture fluid products. So which statement is true with regards of HMF? HMFP can be stabilized using combined hurdle technology. The pH range is typically between 3 to 4.1. High water activity alone is sufficient to inhibit microbial growth. The combination of high water activity and low pH is effective in preserving HMFP. So Suraj is saying C. He is correct. So basically high moisture food products, they can be used and they can be basically stabilized using combined hurdle technology like blanching, water activity depression, lowering of pH, addition of chemical preservatives, which can be like sorbates or benzoates, and the addition of sulfides as an anti-browning agents. The pH can be between 3 to 4.1 of HMFP, which basically might not represent basically the optimal growth of the microorganisms. So the pH basically exerts a strong selective pressure. So because of that, microorganism growth can be inhibited. High water activity basically alone is not sufficient. So there will be a combination of high water activity and low pH so that we can preserve these kind of food products, high moisture food products. At high level of water activity, the effect of the pH on osmophilic yeast might be same as for non-osmophilic yeast. Food is because water activity is high, so their effect can be same. Uh, the food is preserved by interference with the homeostasis of microorganisms by intelligent combination of hurdles. So, 1 is correct, 2 is correct, 4th is correct, and since is not alone sufficient, we should use pH with this. Now let's discuss this question, which is on again high moisture food products. So which of the following methods can be used for their preservation? Use of preservatives, high pressure processing, passive packaging, traditional packaging. C 2, 3, 4. This one. A 1 and 2. Vishakha 1, 2, and 3. Okay, let's see. A is correct answer. B is not. So let's see why. So HMF basically can be stabilized. We discussed about several treatments like blanching, water activity depression, lowering in the pH, additional of different chemical preservatives, and we can use sulfides also. We can use different preservatives, which is true. So high pressure processing can also be used and active packaging methods also can be used. But this is talking passive, third statement is saying passive packaging, which is incorrect, and traditional canning is also not used for the high moisture food products. So 1 and 2 are correct. Next is which of the following is not a novel preservation technique for high moisture food products? HPP, plasma treatment, UV radiation, and open air drying. Vishakha D, open air drying. Suraj is also D. So you both guys are correct. So high pressure processing, plasma treatment and UV irradiation, they are basically novel technologies. Open air drying is not a novel technology, as we can see um, that if we are drying in open air, that is a part of conventional drying, not conventional preservative method, not novel. Novel includes like microwave, radio frequency, ohmic heating, HPP, PEF, ultrasound, irradiation, pulse light, cold plasma, and then ozonation, like that. And conventional uses like freezing, chilling, heating, and acidification, fermentation, ultra like filtration methods, using additives or proteolysis. All these are novel methods, or sorry, conventional methods. Which of the following statement about the processing of fresh cut produces is are correct? The goals of an optimum peeling operation includes minimizing product losses, heat ring formation, energy, chemical usage, and environmental pollution. Second one is peeling methods are manual, mechanical, chemical, enzymatic, and thermal. Third statement is factors affecting the cutting operations include the sharpness of the cutting tools, the type of size reduction, direction of cutting, geometrical shapes of fresh cut fruits and vegetables. The goal of an optimal peeling operation do not include minimizing environmental pollution. Suraj is saying C 1, 2, and 3. Vishakha 1, 2, and 3. Okay, you both are on same. Let's see the solution. 1, 2, and 3. You both are correct. So we have fresh cut produces like can be fruits or vegetables, and basically they are the physically altered form. So it can be like peeled, sliced, shredded, trimmed, or washed form of fruits and vegetables. So initially, in the first step, we receive fruits, vegetables, and inspection is being done. Then pre-cooling and basic cleaning or disinfection. At second step, sorting and grading can be done. At third step, peeling or cutting or core removal is done. And then fourth step, washing and sanitization. And at fifth step, packaging. But before that packing, we should apply hurdle technologies like any thermal treatment, like dipping method, lowering of the pH, etc. So here in this question, we are talking about basically peeling or cutting operation, which is the step 3 operation, this one. So basically peeling or cutting, basic goal is optimum peeling is that the product losses should be minimized, and it depends on the type of the product, like potato products or like that, and minimizing of the heat ring formation in apple and potato, and then minimizing the energy and the chemical usage, and the environmental pollution should be minimized. So this is the basic goals. Peeling methods basically include manual peeling, mechanical peeling, chemical peeling. It can include enzymatic peeling and also thermal peeling. Oh, sorry. So sorry for the disturbance. So peeling methods, basically it includes manual peeling, mechanical peeling, and then it also includes chemical peeling, enzymatic peeling, and thermal peeling. So this statement is correct, second one, and first one is also correct. And the factors which affect the cutting operation, it includes basically the sharpness of the cutting tool, the type of the size reduction like slicing, dicing, or shredding, then which direction we are cutting, longitude or transverse, and the geometric shapes like the in cubes or discs or which form we are cutting. So this statement is also correct. Statement fourth is not correct because it is saying do not include minimizing environment pollution. Rest all are correct. And basically in the peeling or the cutting, we are cutting the basically the whole form of the fruits and vegetables into the cut form. So what will happen when we are cutting? So because of the cutting or the peeling, the outer layer of the, if we are talking about any fruits or vegetables, this is the outer layer. So this outer layer is removed during peeling, and we are cutting, which means we are damaging some of the tissues. So there is both tissue damage and the removal of the outer protective layer. Because of that, the deterioration or the degradation of cut fruits and vegetables is basically more than the whole fruits and vegetables. So, because there is damaged surface area from peeling and cutting, so it increases basically respiration rate, ethylene production, and water loss will be more because there is no peel, and microbial contamination or microbial attack will also be more. And because some of the, like if we talk about whole fruit, if we are cutting, so what is happening, the tissues here, they are damaged. So from the damaged tissue there can be more of the enzymatic action. So overall, in the cut fruit and vegetables, the food safety is a very important factor. And microbial contamination should be minimized so that the overall shelf life can be enhanced and the food safety can be ensured. So in this, option C is correct. Let's discuss this question. Which of the following effects are achieved by applying edible coating on fresh cut produce? So if we are applying edible coating, what is happening is we need to tell that reducing the respiration rate. Second statement is increasing the malondialdehyde and electrolyte leakage. Statement 3 says preserving phytonutrients and discoloration retention. Fourth one is controlling post-harvest decay. Anyone would like to answer? So Prachi is saying C, Suraj is saying B. Okay. B1, 3, and 4. Okay, B, Suraj and Vishakha both are saying B. Okay, let's see. So, yes, B is correct answer. So, we know that there is a concept known as edible packaging, which means that in edible coating, this is basically a primary layer near the fruit. If this is a fruit or vegetable, this is the coating, edible coating layer. So, basically, it is a thin layer which is like thickness is very, very low. It may be less than 0.3 mm, and it can be of any material which can enrobe or which can be near the food, just next to the food product, which can enhance its shelf life. And this is edible, so this can be eaten with the food or can be removed before eating. So the material we are using should be edible, or they should be generally recognized as safe as per FDA. So this edible coating, if we talk about, this is food, and this is the layer of edible coating. So what it is actually doing is, this edible coating layer, it protects the food from various physical, chemical and biological attack. And what happens, it also protect the food from physical, means mechanical stress. And what happens, it also like, since it is a semi-permeable membrane near the food product, so it controls the exchange of the oxygen, carbon dioxide, such gases, and also affects the transpiration rate, which means that the water vapor exchange is also being uh, controlled, or the migration of the water and the gases, the migration rate can be controlled. Better to say diffusion rate, actually. So the diffusion rate of these gases and the water vapor can be controlled by this semi-permeable membrane, which is edible coating. Next is, it protects from the ultraviolet or UV light attack, or any attack from the light, so that there will be less formation of free radicals and the decay will be less. And basically it protects the food from the external or the surrounding atmosphere, so that the shelf life can be enhanced. And this can be made from different components like biopolymers, which as the base material, biopolymers as base materials, which can act as a coating material or a base material. In that, different additives can be incorporated, or different like uh, additives like different plant extract, essential oils, they can be incorporated, so that this biopolymer is basically dissolved in some kind of solution. Like, for example, chitosan can be dissolved in water. So the chitosan can be a biopolymer, and the solvent can be a water. So that can be dissolved in water, and different additives can be added, like maybe you are adding plant extracts like aloe vera gel. All these can be added. And this edible coating can be applied by different methods like dipping. So just making the solution and dipping the fruit directly in the solution and then taking out, and this is the coated fruit. Sometimes with the help of spraying, so just filling the coating solution into some spray bottle and then spraying and drying the fruits, the coated fruits, and get the final coated fruits. And then sometimes with the help of brushing also, so the brush can be dipped into the solution. This is the solution. The brush can be dipped and then can be applied on the fruit surface of the fruits. So this is basically the basic concept of edible packaging. So let's see question. So the edible coating, it basically helps in reducing the respiration rate, because what is happening, it is making a semi-permeable membrane around the food product, which controls the diffusion of these gases. So respiration rate can be controlled, like oxygen, CO2 can be, the diffusion of these gases can be controlled. So respiration rate can be reduced. Then second, it preserves the phytochemical and the phytonutrients. So it obviously preserves the phytonutrient contains and the discoloration retention. So and it controls basically the overall post-harvest decay, but it do not increase the amount of the electrolyte leakage or malondialdehyde. So, like it reduces, not increases. So that is not correct. So second is not correct. First, three and fourth, they are correct. Let's move to this next question. Which of the following statement are correct about antimicrobial packaging? Antimicrobial packaging basically does not affect the growth of microorganisms. Antimicrobial packaging, it can work by killing or inhibiting the growth of the pathogenic microorganism in the food. The absorption mode in antimicrobial packaging releases antimicrobial agent into the food. Immobilization mode allows the migration of antimicrobial agents into the food. Anyone would like to answer? B, C, and D. D. Okay, let's see. So B is correct. Basically, antimicrobial packaging, it works by killing or uh, basically inhibit the growth of the microorganisms, pathogenic microorganisms, which are contaminating the food. So it is correct that it does so. Antimicrobial functions, they are basically can be achieved by adding antimicrobial agent in any packaging material, like in edible packaging material. We just talked about edible coating solution. We can add some of the antimicrobial agents, these chemicals. Natural antimicrobial agents we can add, like some essential oils we can add in the coating solution, and we can apply this. And also we can use this with other polymeric materials also. So first statement is not correct because it is saying that not affect the growth of microorganisms, whereas it is specially added uh, to the other uh, packaging system like edible packaging or synthetic packaging to inhibit the growth of microorganisms. These are the certain antimicrobial agents which is being used in food packaging systems like plant extracts, which includes essential oils or like pomegranate peel extract or some grapefruit seed extract. So fruits, peels or seed extracts can be included in this category. Then fungicides can be used. Then oxygen absorbers like BHT, they can be considered as antimicrobial agents. And polymers like chitosan, they are having this chitosan is a naturally, it is having the antimicrobial properties. Then certain gases like ethanol and also like nanomaterials, like nanometals basically. So sorry, nano, just in like nano, zinc, AU sometimes we use, so they can be used. And then bacteriocins like lysine and all. Then enzymes like lysozymes and organic acids like sorbic acid, propionic acid. They can be used as antimicrobial agents. And certain, we can discuss like certain compounds like silver substituted zeolite. So silver, this can be, this are the basically antimicrobial food packaging system which are used with these trade names. These are commercial ones. So these are the active compounds like silver substituted zeolite is the um, basically active compound in this matrix, and it is used for the like milk containers, paper cupboard cartons, and it's basically, since it is commercial, so it's straight in business. So these all examples which are included in this table, they are commercial one. So one is plant extract like wasabi extract. So it is used as a wasa powder under the name of wasa powder trade name. So one packaging is being made from wasabi extract and PET films so that it can be used. And like chlorine dioxide used with some polyolefins in the films and chassis, and it can also be used as packaging material. So these are the antimicrobial packaging materials. The mode of action of the antimicrobials are release, absorption, or immobilization. So three modes are there. So in release, it allows the migration of antimicrobial agents into the food or headspace inside the packages and inhibit the overall growth of the microorganism. And in absorption, it removes essential factors of microbial growth from the food system and inhibit the growth of microorganisms. Third one is immobilization, does not release antimicrobial agents but suppresses the growth of microorganism at the contact surface. So, since immobilization is not migration, right? It's just at the contact surface. So this one is incorrect. And in absorption, it releases, not it doesn't releases the antimicrobial, whereas it just removes some of the essential parts which are required for the microbial growth. So this one is also incorrect. So only B is correct. Let's discuss this question. What is the oxygen percentage of the sliced orange to develop the beneficial atmosphere in the modified atmosphere packaging? Anyone would like to answer this question? Okay, so first let's understand what is modified atmosphere packaging. So modified atmosphere packaging basically relies on the changing of the surrounding gas. So the composition of the oxygen and nitrogen, oxygen and the carbon dioxide is basically making the modified atmosphere. So it can control the product's respiration rate and the shelf life can be enhanced. So it can be three types: high oxygen, modified atmospheric packaging, controlled atmosphere, and intelligent modified atmo intelligent modified atmosphere packaging. So we can see this is a modified atmosphere packaging. Inside this as a product. So the amount of oxygen and CO2 inside this environment controls basically the modifies the atmosphere and controls the shelf life or the respiration rate of the food product. If we see from this table, so which is already discussed in the lecture slides, so we can see that sliced orange, it is having around 14 to 21% oxygen and 7 to 10% CO2 in modified atmosphere. So option D is correct in this case. Is this question clear for everyone? So this was the last question. So we discussed basically today the minimal processing of food products, like we discussed about novel technologies and different packaging methods. Then we discussed about the hurdle technology concepts, its types and effects, and also the different hurdles like the pH water activity and the temperature on how it is affecting the microorganism and how microorganism is uh, basically trying to maintain the homeostasis and different mechanisms how these different hurdles act on a microorganism. Then we discussed about non-thermal hurdle techniques, and then we discussed about intermediate and high moisture food products, their production and processing and preservation, and different novel technologies for their preservation. Where then we discussed about cut fruits and vegetables and their manufacturing, different packaging methods, like we discussed about edible packaging, modified atmosphere packaging, and antimicrobial packaging methods. And then in the next week, we'll be discussing about um, juice extract and clarification, and we'll be discussing about concentrates and pastes, aseptic processing and packaging in detail, and then RTS and RTD beverages, and then lastly the quality and safety aspects. So this recorded lecture will be uploaded on this YouTube link, and the PPT will be uploaded on the Google Drive link. And if you have any doubt, you can write to me or you can ask on Q&A group. So thank you so much for joining. And if you have any doubt, you can stay in the meeting, or otherwise we can meet on next Sunday, which is on 7th September. So thank you so much for joining today's session. If you have any doubts, you can stay. Otherwise, you can leave the meeting.